Initiation of the alloimmune response takes place when alloantigen-specific T cells come into contact with professional antigen-presenting cells expressing or presenting the alloantigen recognized by the T cell. This contact mainly occurs in the secondary lymphoid organs of the recipient. It is this cognate interaction which triggers T cell activation. The T cell receptor specifically recognizes the alloantigen in the form or in the context of MHC class I or class II expressed on the antigen-presenting cell, thereby receiving signal 1. However, for full activation, the T cell should also receive signal 2 or co-stimulation. CD80 and CD86 on the antigen-presenting cells cross-link the CD28 receptor on T cells. Together, signal 1 and 2 result in efficient signaling through the T cell receptor CD3 complex, inducing several signaling cascades, including a rise in intercellular calcium. The interaction of calcium with calmodulin will activate the phosphatase calcineurin. Upon activation, calcineurin will dephosphorylate the transcription factor, nuclear factor of activated T cells, or NFAT. Upon dephosphorylation, NFAT can translocate from the cytoplasm to the nucleus and bind to the promoter region of the interleukin-2 gene, amongst others, resulting in an enhanced transcription of this cytokine. Increased expression of interleukin-2 mRNA will result in translation of the interleukin-2 gene into protein and secretion of interleukin-2 by the T cells. Secreted extracellular interleukin-2 or IL-2 binds to the IL-2 receptor and induces a signaling cascade of various kinases, including the PI3 kinase pathway and the molecular target of rapamycin, or mTOR pathway, which initiates the cell cycle. Lymphocytes require synthesis of purine and pyrimidine nucleotides for replication as building blocks for DNA. T cell activation results in proliferation, which leads to a large number of T cells with identical specificity.